followed that up with 2 master's degrees, always paralleling computer science and biology throughout her career, really embodying this idea of an interdisciplinary scientist. She's now a professor at University of Colorado and is going to talk to us today about eRNAs and how you can use them to learn more about your favorite transcription factors. So with that, let's give Robin a round of applause.
Thank you, Phil. And thank you for the invitation out. It's been a long time since I've been in Vancouver. I was actually a graduate student at WashU the last time I came here. I was trying to remember for which meeting, but that's a whole other story. So I want to talk today about a problem that we study a lot in the lab, and that is transcriptional regulation. Our species is extremely diverse and differs very small between any 2 people within this room. But those differences obviously lead to what we believe are large phenotypic distinctions. They also lead to disease, and many diseases that we're interested in are actually mutations within particular transcription factors, including p53 itself, a transcription or tumor suppressor that is mutated in about half of all cancers. In order to understand mutations in transcription factors or to understand the mutations in the places the transcription factors target, we have to have a better understanding of regulation. Because in fact, when you look at the output of most genome-wide association studies, this number varies depending on where you look it up, but somewhere in the order of 3/4 of all disease-associated variations fall within these regulatory regions. So I always hate central dogma slides, so I will be quick with this. Nobody in this room needs to be told that DNA makes RNA, makes protein, which makes cells, which makes you, and we're not going to get any further than just that first arrow today. We're going to be interested in transcription itself, and transcription itself is very easily and well described. You have some really important gene, and it is transcribed into making RNA. These transcripts and their regulation arise from 2 different types of gene elements: promoters and or enhancers. And promoters are enhancers, and my clicker just died, are these guys that are upstream. Promoters typically being proximally upstream, enhancers being somewhere located within the genome, doesn't even necessarily have to be close by. And these particular regulatory elements, when present or activating a gene, give rise to increased expression in that gene, or transcription in that gene. So how does this actually work sort of mechanistically? The way that this is usually described is some particular regulatory element has associated with it transcription factor binding sites. Transcription factors, they come down, they bind to these enhancers and or promoters, and in so doing stimulate the production of RNA polymerase through the gene, giving rise to RNA. So how do we study this process? Generally speaking, we study this process by focusing on either those transcription factors or focusing on RNA polymerase itself. And so we can study transcription through things like RNA-seq or microarrays, or we can study transcription factor binding through processes like CHIP. And typically, to understand where a transcription factor binds and how that modulates transcription, we're going to take our favorite transcription factor, and we're going to either overexpress it, or, sorry, I'm directionally challenged, overexpress it or knock it down, and then ask how these changes, and apparently you can't touch that either, these changes influence that expression. And we may do the exact same sort of knockouts and overexpressions in CHIP data. And all the while we're trying to decipher from this which transcription factors are important for binding to which regulatory elements, and how do those then control particular target genes. So for the course of this talk, we're going to use one particular model system. We're interested in the activity of p53. That's that tumor suppressor I mentioned earlier. p53 is controlled by MDM2. MDM2 binds to p53, and in so doing prevents it from regulating gene transcription. There is a small molecule inhibitor of this interaction known as nutlin that actually binds in the interface of MDM2, and in so doing stimulates p53 activity. So we're interested in what are the direct targets of p53 and what genes does p53 regulate. But I'm going to argue that the way that most people study these kinds of questions is in fact wrong. Most people approach this by starting with the RNA. Take your cell, pull out all the RNA, activate p53, pull out the RNA, and ask which ones differ. And the problem with this is that is not a representation of transcription. RNA is a function of what's being transcribed, but also a function of what's being degraded. And so any changes in steady state may be changes in transcription, but they could also be changes in steady state RNA. So we need a different approach than your classical RNA-seq if we really want to ask questions about transcription. And so my lab spent the last several years really focused on nascent transcription assays, one of which is global run-on sequencing. So briefly, how does GRO-seq work? GRO-seq works by you have transcription occurring genome-wide at lots of different places. And you want to pause those polymerases. And you do so by basically isolating nuclei in very cold temperatures. That starves them in MTPs, and the cold slows down the polymerase process and pauses things. Then you restart polymerase by exposing it to a number of NTPs, labeling one of those NTPs, in this case UTP, with bromine, and that will allow polymerase to pick up, goes, okay, I got to keep moving, moves along, incorporates those NTPs into the production of the transcript, and then you can use those incorporated labeled NTPs as a way to pull out those transcripts that are just coming off the back of polymerase. So you do a short pulse of the run-on, about 5 minutes. Polymerase will proceed about 100 to 150 bases, and it gives you a little tag to tell you where was polymerase at the time you did this experiment. You then sequence this. That gives you a whole bag of reads. You map those back to the genome, and you get a profile that looks like this. So I'm going to show a lot of these profiles. The x-axis is some coordinate in the genome. Generally speaking, blue is the positive strand, red is the negative strand. And in this particular case, unfortunately my pointer still is maybe working again, polymerase is proceeding in this direction in the blue strand and towards me on the red one. And the height of the bar is the number of times we recover that read, and that is proportional to the transcriptional activity, or how many times did we polymerase in that location. So going back to our experiment with Nutlin, we take Nutlin, we expose cells to that particular chemical to activate p53, and we have both a pre and post Nutlin exposure. And after Nutlin, we see this dramatic increase in bidirectional transcription, and in fact, the vast majority of those bidirectional transcription sites originate from p53 binding locations, which is a little bit of a conundrum. How do you get a transcription factor that binds to double-stranded DNA and the production of transcripts essentially localizing to the same location? Well, it turns out these little transcripts have come to be known as eRNAs, or enhancer RNAs, because many of the binding sites in the genome are located within enhancers. And we became very interested in identifying where these enhancers are, or eRNAs, and what they do. So here's a typical gene in the genome. GRO-seq, or RNA-seq, gives you, you've all probably seen assays like this, a very stereotypical exon-intron boundary response. GRO-seq gives you a very different perspective. You are pulling those little BRUTP marked molecules out of the back of polymerase as they're being produced, so they're actually pre-splicing, even though splicing is co-transcriptional. And so you get a very different perspective. You get a bidirectional transcript at the 5' end, followed by elongation through the gene without evidence for exon or intron boundaries. In addition to seeing this at annotated genes, you see lots of bidirectional sites of transcription away from annotated genes in regions that we now know to be marked with enhancer marks. In RNA-seq, the height of the bar is roughly the expression level, or mRNA concentration. It's a steady state assay. It's a function of transcription and degradation. In GRO-seq, the height is the number of times you see RNA Pol II there, and is really a direct measure of transcription itself. And we want to come up with a method that can analyze data sets like this one and identify all of the transcripts therein. And so I'm going to tell you about a couple of different directions we took at this. The first was simply to realize that in places where we see transcription, we expect to see sort of contiguous read coverage, more or less, through a large region. And so we want to take GRO-seq reads, in this case I'm only showing one strand, and segregate those into regions of active transcription from regions of inactive transcription. And because of mapping biases and other noise type processes, you will always in the inactive have a small number of reads. And because of lack of sequencing depth and or also mapping issues, you will always have little gaps within your active regions. And the idea is to identify which regions are active and which regions are inactive. Simple segregation kind of problem. We can take our reads, map them to the genome, and then identify, hopefully, active regions as having a lot of reads mapping and inactive as having rare read mapping. So the real way of thinking about this that we started was that actually what we were looking for are regions of contiguous coverage, and therefore you're looking for contigs in the traditional assembly sense. So we would go through and say, okay, we can assemble the reads we map for GRO-seq into contigs, and we expect in regions of high active transcription to have long contigs and short gaps, and in regions of inactivity to have short contigs and long gaps, which I'll arbitrarily label as a negative. We also have length, we have depth coverage, we have a number of statistics that we can describe for any one of these contigs or any one of these gaps. So I can take that pile of features and start to ask which of these features segregate an active region from an inactive region. So in this case, I'm now looking at gap lengths, and my expectation is that very small gaps like this one are actually more likely to occur within active regions, and very long gaps are more likely to occur within inactive regions. And rather than segregate on individual criteria such as this, I can feed in a number of these criteria into a Markov model framework, basically building a maximum entropy Markov model that separates on multiple different dimensions at the same time to identify the active from the inactive regions. We called this little package fstitch, and here's an example of fstitch's output. For this particular transcription region, we run it independently on the positive strand or blue strand and then separately on the red strand. And it labels in the boxes at the bottom, blue regions as regions it called as active on the blue strand, red regions as regions it called of active, and green in general is those that are off. And of course, because it's a probabilistic model, we get an associated probability score for each of those labels. So fstitch taught us a few things about transcription. But before we get to those, we wanted to make sure that, this is a trained maximum Markov style model. We needed to know how sensitive it was to its training. So it turns out that fstitch works really well, even with very small data sets. We started with the entirety of chromosome 1, actually annotated by hand by an undergraduate in the lab. It was quite a task. And then we just subsampled subparts of the chromosome until we got down to where we realized that you sort of broke down when you had anything less than about 5 on and 5 off regions. So it actually does really well with very small amounts of training data. We also started with what was at the time the most deeply sequenced data set that we had and asked how much does the sequencing depth, this nascent transcription, influence what you can detect. And you could go a lot shallower than that particular, this was one we sequenced, which of course was not reassuring to our pocketbooks, but hey, that's okay. But you could go a lot shallower than that data set and still recover most of the regions. But what did we learn from fstitch? So the first thing we learned is we took all of the annotated genes and we aligned our regions of GRO-seq signal to those annotated genes. And we noticed a couple of things. One, everybody already knew that there was a robust peak at the 5' end in transcription because polymerase loads there, and the loading process is slow, and there's an initial pausing. What was less well appreciated was this strong and robust peak at the 3' end of genes, suggesting that in the process of cleavage, because this tends to occur at actually the cleavage site or the annotated end, there appears to be a polymerase pausing event. And polymerase proceeds much further past, as shown by the hash marks on the right there, that cleavage site. So the site that we annotate as the end of the gene is the cleavage site and the polyadenylation site, and polymerase proceeds past that some amount. So how much does it proceed past that? Well, at the 5' end, most of the polymerase loading is pretty close to the annotated start. Here is the 5' end estimations for annotated genes for our method in green, as well as a windowing method in blue, and in red, a method that was actually another hidden Markov model, but this one was trained directly on annotation. And so being trained on annotation, it basically gets the annotation and not what the data tells you. At the 3' end, however, we saw something distinctly different. and that is that polymerase proceeds past the cleavage site sometimes as much as 25 or 30 kb. This is actually well appreciated in the biochemists that study polymerase and polymerase processivity, and they've wondered for a long time what exactly is the signal that causes polymerase to actually stop. And that's fairly contentious, but we're hoping that some of these data sets in GRO-seq will help with that question in the future. So in our p53 experiment, we noted this robust activation of eRNAs over p53 binding sites. And it made us wonder whether or not this massive increase in transcription, differential expression observed at p53 sites, meant that you would be able to take this p53 data set and infer that it was actually p53 that was driving the transcription. So this is a pretty commonly done thing. You take all the sites of things that are differentially expressed, you pull them out, and then you do motif finding. And our expectation is that we would identify p53. And we were hoping, and it's interesting that that shows up as black on here. It doesn't do that on mine. It's a beautifully multicolored motif, but that's okay. We were hoping we'd recover the p53 motif. This is like your last sanity check on your data. And in fact, it failed miserably. Okay? And the reason it failed miserably, I hope, is demonstrated here. And that is that F-stitch would call this region as on because there are, in fact, blue reads contiguous throughout the region. But that's not what your eye sees. What your eye sees are multiple distinct peaks within the blue or peaks within the red, suggesting there may, in fact, be multiple very tightly spaced transcripts in this region. So despite the fact that F-stitch was very useful, and it was a great first approach to this data set, we needed to do something different. And so we got to thinking, what actually are the biological molecular processes underneath GRO-seq? So GRO-seq is pulling nascent transcripts as they're being produced. It's completely agnostic to which polymerase is driving those, so we'll pull transcripts from Pol 1, Pol 2, Pol 3. But when you map them back to the genome, the vast majority of all what you get is Pol 2 transcripts, and that's because of repeats in Pol 1 and Pol 3 transcripts. But Pol 2 and the way it behaves is very well understood. So Pol 2 has 4 distinct stages of activity. This is a massive multi-unit subcomplex. First, it loads. It picks some location in the DNA on which to load. That loading process does not produce a transcript. We cannot see it by GRO-seq. It then picks a strand. That strand selection seems to be somewhat random and arbitrary, surprisingly. Once it picks a strand, it melts the DNA, separates the 2 strands, and proceeds to produce a short abortive transcript and pauses. These short abortive transcripts can be on either strand depending on where that initial strand selection was chosen, but now we have a transcript that GRO-seq can pick up and see. A subset of polymerases make it past this pause point, and they proceed in a unidirectional fashion through the gene, producing the full-length gene transcript. This sort of stereotypical behavior of Pol 2 is reflected in the GRO-seq data itself. Again, we don't see loading. We only see strand selection because we have a stranded protocol and you see positive and negative strands, but it's not explicit to any particular location. But at the point of initiation where you get these bidirectional abortive transcripts, you see bidirectional transcription within the GRO-seq reads. Red and blue running away from each other in a very close proximity. We see this at all annotated genes as well as these little eRNAs randomly throughout the genome. So we think that eRNA production is simply the short abortive transcript aspect of polymerase behavior without ever transitioning into elongation. When you do transition into elongation, you get complete gene length transcripts. So with this kind of a description, we should be able now to describe this as a mixture model. So loading, we're going to pick some particular position with some variance, and that we're just going to describe as a normal distribution. We can't see loading, so we don't actually ever get that distribution on the far right, but if we could, that's what the CDF would look like. Once we load, we're going to pick a strand at random. This is akin to a Bernoulli. It's not quite a Bernoulli because Bernoullis are described as 0, 1, but in order to do coordinate math, it's actually better here to describe it as -1 and +1, but we're going to pick a strand. Once we've picked a strand, we're going to proceed in a 5' to 3' fashion only with an exponential decay. This is finally the first process you actually see in the GRO-seq data, and it produces within the GRO-seq data that bidirectional transcript you see at the far right, where that tends to be a structure. And a subset of these polymerases will do initiation or loading strand selection initiation and proceed into elongation. As they elongate, the rate of polymerase activity is roughly semi-fixed, and so you get something that resembles uniform with high variance through that region. So you end up getting a distribution in your GRO-seq like you see at the far right, similar to what we see at annotated genes. So you can now describe all of GRO-seq by a convolution of these small mixtures or these processes. And that convolution, if you generate reads from it, produces data that looks an awful lot like real data, which made us feel like maybe we're on the right track here. But unfortunately, being able to generate from it, which is actually quite trivial, is not the same as being able now to take raw data and fit it to the model. And so you need some way to take raw data and fit it to the model. And I'm gonna scare you only briefly with something that is very mathematical, but there are a lot of things we don't know when we go to fit this. And one of them is how many stages or how many components or mixtures do we need to describe the data? Where do we expect loading to occur? These are the sorts of things we want to infer when we do this fit. So this is what the likelihood function looks like. I assure you there's no analytical solution, unfortunately, even though I had a student bang their head against a glass wall for a while looking for one. But if you knew either the position polymerase loads, that's X, or Y, which is the link that processes for that initiation, then you could analytically solve it. Not knowing both of them, however, makes it an underdetermined problem. But the good thing is probability can come to the rescue, and you can compute conditional probabilities for either X or Y or both, and use those conditional probabilities in a classical EM algorithm and estimate all these parameters. So here's how it works. Here's a region of real data. The black lines are the algorithm as it's working through a number of iterations, and you'll see as it progresses, is slowly going to lock in and find essentially where that normal, that loading, those exponential decay and the elongation is. It also works fairly well in regions that transcription is dense, and here it's going to lock in and find 3 distinct loading locations and 6 transcripts. So we packaged all this thing up in this thing we call Tfit. It runs an OpenPI on very large clusters. And you can feed it GRO-seq data, and it outputs essentially a whole slew of parameters about every transcript. Those parameters include things like, I don't know why that's not showing up, but include things like the position of polymerase loading, the variance, the footprint, or how far apart are those 2 bidirectionals, the strand bias, how far does it initiate before it pauses, the pausing ratio, elongation, etc. We see changes in every one of these parameters between data sets dependent on particular perturbations. But for the sake of the rest of this talk, I'm only going to focus on mu, or the position of polymerase loading. So our working hypothesis is that a transcription factor binds to DNA with some recognition motif, and in so doing recruits polymerase to that location and gives rise to one of these bidirectional transcripts. The question we wanted to ask then is, if I give you the bidirectional transcripts, can you infer the set of transcription factors that actually had to have given rise to them? It's a $1 million question, one everybody in studying transcription has been trying to do for 30 or 40 years, right? So we went back to our Nutland experiment. And the idea is quite simple. There are a number of p53 motifs distributed randomly across the genome. If I look at my control sample, my DMSO, p53 is not active. I expect to see some number of positions of polymerase loading, those represented in green on the far right there. Those are distinct from the positions I will see when I expose it to Nutland. And if p53 has been activated between those 2 conditions, my expectation is that I will see a correlation between the position of p53 and the position of polymerase loading under this model when p53 is active. And in fact, that is what we see. So on the left is a histogram of the position of p53 motifs relative to eRNAs in a condition where p53 is not active, namely DMSO. I can represent this as a conventional histogram, or I can summarize it into that heat map at the bottom. The center of this histogram is the middle of those bidirectionals, or the loading position of polymerase genome-wide. On the right is the distribution of p53 motifs relative to eRNA midpoints in the case where p53 is activated by Netlin. And you see this very stark contrast. You see this very sharp peak right at the position of polymerase loading. This suggested that if you could quantify how much of the heat, so to speak, in these histograms is proximal to that polymerase loading position, you could identify which transcription factors were active simply by those that had high co-occurrence from those that showed random barcode patterns. So does this work? Well, here is one particular cell type, K562s, and the distribution of motifs for all 641 motifs in a motif database. And what you see is we have distinct clusters. There are certain subgroups of motifs that really show this co-localization centering pattern. The motif tends to be right at the beginning of an eRNA. We see subsets of motifs where there appears to be no enrichment in that random pattern we expected. Interestingly, we also see a set of motifs that look depleted, or absent from the places where we expect them more often than by chance. And we see this weird group of guys that are flanked or displaced at some distance from the eRNA origin. And in order to quantify these patterns, we just developed a really simple statistic. It doesn't get much easier than this. We just asked what fraction of the heat is within 150 bases of that place of polymerase loading versus a larger localized window. That we called an MT score, motif displacement. We can calculate that MD score for any transcription factor binding motif for any single GRO-seq data set, and that effectively allows us to resort this data set. I'm going to jump to the punchline at this point and tell you that the ones in blue, we believe, are predictions for the transcription factor set that are active in this cell type. The ones that are in orange are also transcription factors that are active in this cell type but are repressors. And they're actively repressing the presence of eRNAs, which is why you get the depleted signal. The guys in black, the ones that are off-shifted, turn out to be a really interesting case we haven't fully got our head wrapped around. A subset of them are nucleosome modifiers. The other subset are conventional transcription factors that will be offset in one cell type and not offset in another. And that's giving rise to some very interesting transcription factor interactions. So the way to think about these MD scores is that the motifs in the genome don't change. They're randomly distributed. So I can take any given genome, calculate my expectation if I randomly sort eRNAs for any given specific motif. Motifs have some GC-AT bias. Turns out enhancers and promoters are very GC-rich. And so your expectation on your MD score is quite variable. For a given data set, here shown on the y-axis, I can then calculate my observed MD score, which reflects where the eRNAs are currently present. And when that observed is enriched relative to background, the red, those are the guys that are active. When it's depleted, those are the guys that are active as repressors. So I can take this active and depleted, now I have to say, how do I validate that this is the set of transcription factors that are actually working in this cell type? That turns out to be really, really hard problem. The first impulse everybody has is to compare it to transcription, and in fact, GRO-seq is a transcription assay. So first thing we do is we say, okay, I'm going to take all the red dots, and I'm going to say, is the transcription of the transcription factor gene itself elevated in the red and green dots relative to the blue ones? Well, the problem is that no single transcription factor is both red and blue in the same experiment. So I have to look across multiple experiments. So we downloaded the 770 GRO-seq experiments that have already been published. And when you do this for the red set, you see transcription is only marginally elevated for the set of things that we call active in the cell types in which we call them active. And the set that we call depleted is only marginally elevated, again, relative to the set that we call at expectation. So first, this seems like it's at least consistent, but it's not an overwhelmingly endorsement for our hypothesis. But then again, if this worked, this would have been a much easier way. I mean, we would have figured out how to predict which TFs were active 35 years ago. And take again P53 as an example. P53 is not regulated at transcription. It is not regulated by post-transcriptional modifications. It is regulated by its interaction with MDM2, which means transcription assays, proteomics assays, even phosphoproteomics assays are not going to tell you whether P53 is active. So how do we test whether we know we've got the active subset of transcription factors? We have to go to experiments where people have directly targeted individual transcription factors for activation. So our Nutlin experiment is exactly one of those experiments. Nutlin is an exquisitely specific activator of P53. How specific? That was another question we could ask. So we can take GRO-seq, before and after exposure to Nutlin, and now take that same database of transcription factor motifs and ask how do they change in the presence of the drug. Now the x-axis is the number of eRNAs responding, and the y-axis is the change in MD score pre and post-drug. For a transcription factor to change in MD score, because the motifs are not changing, it must imply the presence of new eRNAs being driven by that transcription factor. And in this case, the only two TFs that rise above and change in a significant fashion are P53 and P63, which have nearly identical motifs. This is not specific to P53. I can expose cells to TNF-alpha. TNF-alpha is known to activate NF-kappa B1, and in fact, when we do this exposure, we recover by MD score the 5 subcomponents of NF-kappa B complex. I can also expose cells to estradiol, known to activate estrogen receptors, and when I do this, I get the estrogen receptors that go up and a number of fork heads that go down. And interestingly, about the time we published this, we were looking through the literature saying, what the heck is this stuff that goes down? There were no reports of transcription factors that get turned off by estrogen. And about 3 weeks later, one of my students comes in with a brand new published paper where somebody had showed that, I believe it's FOXD1 and not FOXF2, had been shown to be an absolute required for estradiol response. If you knock it out, the response to estradiol is suppressed. We don't know about the other ones. We're still chasing those. But it looks like in cases where people have taken known activators of specific TFs, we can infer which TFs changed, having not known what the activity of the drug was, suggesting we at least have some traction on being able to identify the active subset of transcription factors. So now I can take my transcription factor, my favorite, and ask over the 400 or 500, in this case 491 human GRO-seq data sets, what is its MD score or activity profile across all of the data sets? And you see it's pretty dynamic, but there are some subsets in which the MD score is significantly higher than others. The question is, what do those experiments have in common? Many times we're finding that what they have in common is an underlying cell type. So here's an example. NANOG has an expected MD score, and when I look across 500 experiments, I can now calculate the change in MD score relative to that expectation, and you get a distribution that's largely centered around 0, meaning most things NANOG is not actually on and functional. But you see a little bit of mass out to the right, and the question is, what does that mass have in common? Well, it turns out all of those are embryonic stem cells experiments. That's reassuring. NANOG's a marker for embryonic stem cells. You see similar things with GATA1, a marker for leukemia, and in fact, we only see activity in leukemia cells. Applying this to all transcription factors and now looking for cases where a particular transcription factor is specific to a subset of cell types gives you a network diagram similar to this, where for different cell types, I have different numbers of cell type specific transcription factors, and in some cases, subsets of cell types or subsets of transcription factors that are only active in subsets of cell types, allowing a new metric of sort of how similar are two particular cell types. So we think we have a new approach to at least giving us some information about the activity of transcription factors. We're only able to do this because we have a model of what GRO-seq data looks like that's based on a biological phenomenon, meaning the behavior of polymerase. And from this, we're now getting an unprecedented view on transcription factors and what they do, and how to tie those to things like which isoform is generating that data and who does it interact with. And with that, I'll take any questions. Yes.
Very nice presentation. I'm coming from different kind of lookout.
Yep.
I want to just check if there's some possibility to actually select those responsive or chemoresistant response to kind of transcription factors, and who are its targets that they actually down-level regulate, suppress, or activate. Is it possible to actually select those transcription factors and see actually which eRNA?
So I didn't catch the modulator on which type of transcription factors?
Yeah, I mean, the significant one from the
Yep.
whole analysis.
Yep.
And those that actually target the significant or genes or
Yep.
proteins that actually deregulate or activate that kind of thing.
So we've now,
Let's see if one transcription factor actually responsible for a multiple gene up or down regulation controlled by a pathway,
yep.
so that we can target the pathway for the
Yep.
resistant.
So we've now exposed cells to a number of different drugs. The timing after exposure at which a particular transcription factor becomes responsive depends on the drug. So in the cases that I showed in this talk, all of these drugs are known to activate particular subsets of transcription factors or subunits to a larger transcription factor complex. Other drugs, such as cortistatin A, targets mediators everywhere. And what we see in the case of cortistatin A is that the initial transcriptional response you see at 25 minutes or 30 minutes or 40 minutes is changes in the pausing ratios within GRO-seq. In other words, how much of polymerase is at the 5' end versus elongating through the gene. But interestingly, at the 45-minute to an hour point, suddenly targeting mediator results in a change in a number of different transcription factors. And we have phosphoproteomics data to support that that's because one of the components of mediator is a kinase, and cortistatin A appears to inhibit that kinase activity, and that kinase activity usually regulates those transcription factors, which is a long-winded way of saying that temporally we can sort of order events in this fashion as long as you have the GRO-seq for the right time points. We've done short-term time points as short as 12 minutes. With estradiol, you can see changes in estrogen receptors as short as 10 minutes. With other drugs, it's 25 minutes to 30 minutes before you start to see a transcription factor change. And we've done circadian rhythm and infection time points that go out to 24 hours. And for example, in the infection time point series, you can actually see in the data that NF-kappa B is an initial responder and the interferons are a late responder, which is, of course, consistent with the literature, but we didn't have to know that when we did the experiment.
That's how because you have this user-friendly tools like RNA,
Yep.
that give you all these predictions on showing your input, goes in encode database,
Yep.
all the chip-sheet data that they have, and all this.
Yep.
Gets out into all this kind of network and gives you a network of one transcription factor and your predicted genes around.
Yep.
There's not really the answer.
Yeah, so we've done a lot, and I didn't talk about this at all here, but we've done a lot with comparing nascent transcription data to ENCODE. And the short answer is that only a subset of transcription factor binding sites actually have eRNAs associated with them. But if you knock out a transcription factor, the subset with the eRNAs are the set that respond in the knockout, suggesting that many of the binding sites that you detect by chip are actually non-functional or silent with respect to transcription, and that for whatever reason, the eRNAs are a marker for the subset that is not silent. And so the nice thing about all of this stuff that I showed tonight, and most of the work we've been doing, is that means that we can drop the chip out and still get essentially the signal back, which is pretty cool. Any other questions? Everybody's just starving, right? That's fine. Do you have a question? Yes.
So it's super fascinating. So is that most of the kind of cell lines into a single drug? So we know how.
Yes. So I don't think there's a microphone picks up.
So I should repeat it? Okay.
Yeah. So lines that you can cell lines that you can treat by single drugs. So how can you speculate how this would look if you're looking at like bulk tissue samples?
Can you speculate as to how this would look if you were looking at bulk tissue samples? So there's the pessimist and the optimist view of that. So most tissues are heterogeneous environments in terms of cell type. And cell types clearly have distinct enhancer profiles. And so you'd be getting a mixture of those different cell types. And there's been some really nice work by others with ATAC profiles showing that you can, in a mixture, if you know what the individual cell types within the mixture should be, decipher how many cells of type A, B, C, and D you had. So you could imagine a scenario where you could take a technology like that with a drug and be able to get back what does the drug do on each of the constituent cell types. That's the optimist perspective. The pessimist, or maybe the realist answer to that, is that unfortunately GRO-seq is kind of a nasty protocol. It's a very long multi-day thing. Multi-day is an understatement. If you did 8-hour days, it would take 15 days to do GRO-seq. It also takes a lot of cells at the front end to make it work. And so consequently, there's not much primary tissue or primary cell of any type for which people have done GRO-seq on it. We've done a lot to optimize that protocol. We can now do GRO-seq in about 4.5 days. And because of those optimizations, we can now do GRO-seq in a number of primary tissues. But what we're finding is that primary tissues are extremely variable in how transcriptionally active they actually are. I mean, the nice thing about cell lines is they're basically broken and they transcribe everything all the time. And so you get a really robust GRO-seq signal. But like, for example, quiescent B cells, they transcribe, and this is an exaggeration, but they transcribe like 2 transcripts every 3 days or something because you need astronomical numbers of them in order to get an experiment to work. So the pessimistic answer is the odds of you getting to GRO-seq to work in certain types of primary tissue are going to be pretty low anyway, unfortunately. Yes? Yep. Yes. Yes. You're in reference to this guy. So there's a small subset of those with the offset pattern that are chromatin modulators. Usually they either lay down or remove a particular histone mark. And that makes some amount of sense because eRNAs tend to be in open chromatin regions, and therefore the histones are offset from that open chromatin region, and the eRNAs are being produced from the middle of the open chromatin region, and I would expect any transcription factor that's laying down a histone mark should be offset. We felt really good about that answer until we started to see other cases. And in particular, I'll show you one that I find fascinating, and that is RUNX1. So RUNX1 is a transcription factor that is mutated in a lot of individuals with leukemia and familial platelet disorder. And when we call it as active, we tend to see higher transcription of RUNX1, and when we call it as inactive, lower. This is all sanity checking. And we see it in the right types of cell types, B cells and AC16s, which is a cardiac blood cell. But when we look at the motif displacement distributions, we see 3 distinct patterns. We see in the B cells and T cells that center thing that we would call as active, which is beautiful. We see the inactive pattern, which is great. And then we see this really weird offset pattern in things like bone marrow and embryonic stem cells that we really can't explain very well yet. If you look around 0 in those displacement profiles, they're actually very strongly enriched for another transcription factor called PLAG1, which has very limited reporting in the literature as having any relationship with RUNX1 at all. So we don't know if it's PLAG1 or something that binds to a motif similar. More interestingly, though, is because we have GRO-seq data, we noted a second trend, and that is that RUNX1 essentially comes in 3 major isoforms, and when it's in the centered form, it's always being transcribed from the first 5' end and producing the long isoform of the transcription factor. And when it's got the offset pattern or is inactive, it's being transcribed from the short form. So we think that this offset pattern is some kind of an alternative form of the transcription factor itself and is likely interacting with somebody that's actually at the epicenter of the 0 point. But I'll note that that offset is about 400 bases, so it would be an unusual interaction. And we're still trying to figure that one out. But it's a really great question. And it's not the only one that does it. Yes, ma'am. Why are the eRNAs transcribed? That is a million-dollar question right now, actually. So it is well understood that eRNAs and the target genes of that particular enhancer are transcribed, and in fact, the transcription levels between an eRNA and its target gene are correlated. If you knock down the eRNA by an RNAi or a locked nucleic acid type approach, you actually reduce transcription of the target gene quite significantly, and you could reduce detectable 3D interactions by things like 3C, 4C, 5C, high C, chia-pet. So there is some sort of a spatial interaction that's going on here, but how the transcript, i.e. the eRNA itself, is important to that interaction is really unclear. There's a lot of theories, some of them having to do with just segregating polymerase or recruiting polymerase, some of them having to do with potentially recruiting the transcription factor itself. Many transcription factors actually bind to RNA just as well as they bind to DNA. Some of them have to do with titrating off NELF. So negative elongation factor is a component of polymerase. It's really critical for polymerase loading, but has to be lost for polymerase to transition into that elongation phase. Well, NELF turns out to be a very good non-specific RNA binder. So one of the theories is that if I just need to produce a bunch of little short RNAs in a specific region, that will cause NELF to preferentially jump to the RNAs and fall off. So there's lots of theories, including that it's just noise, and there's very little evidence for any of them. So I think it's still very much an open question. And as far as I know, no one has looked at whether eRNAs are enriched for secondary structure. We have talked about doing it. I actually did my graduate work in Sean Eddy's lab doing RNA secondary structure predictions. You'd think this would be something we'd already tackled, but it turns out my interest and getting a student interested in are very different things. So, all right, thank you guys.